The first questions I'll ask are around sourcing and starting materials. Kathryn, since you're a manufacturing executive, have you start us off. For those who don't know, bit.bio makes induced pluripotent stem cells. So Kathryn, if you wouldn't mind sharing with us what cell type bit.bio uses as a starting material and what you think are the important factors to weigh when selecting a source of starting material.
Great, yeah, thanks for having me. So bit.bio makes iPSC derived cell therapies, and we also make commercial research products that are derived from iPSC cells. Generally, we start with either fibroblasts or cord blood derived cells or similar cells. We then use a variety of the available options to make those into an iPSC. And then from there, we have a technology called Optiox, which utilizes genomic safe harbors and transcription factors to basically drive those iPSCs towards a specific cell type. So currently we have about 27 different products on our commercial research lines, everything from microglia to different types of neurons, and a variety of other types of cells. And then our clinical work initially is going to be focused on the liver.